In this video, we're going to start to look at how we digest lipids. Now, triacylglycerols are water insoluble, while our enzymes that work on them are water soluble. So that's a problem. So all digestion of lipids happen where lipids and water actually meet. So the rate at which we can digest lipids is dependent on how big this interface is. And this interface can be increased by the churning movement of your intestines and the emulsifying actions of different bile acids. So a bile acid or bile salt, every time you hear that word, it's just a amphipathic detergent-like molecule. Remember, amphipathic means polar and non-polar. And they help to solubilize these fat globules by putting them into micelles. And these look like, these bile acids, bile salts, they look like cholesterol derivatives. And they're made by the liver and stored in the gallbladder. So this is their basic shape. Based on what you have the R1 and R2 positions, you get different names. Now, once you eat fatty acids and you want to digest them from the gallbladder, they'll be secreted into the small intestine. And this is where the majority of your lipid digestion and absorption takes place. Now, the specific enzymes that are responsible for digesting these fatty acids starts with lipase. And here you're going to hydrolyze triacylglycerols at the 1 position and 3 position. And this activity, as we said in the previous slide, it will happen at the lipid-water interface. And so the enzyme, the activity in the enzyme actually increases once it hits this interface. We call that interfacial activation. I'm going to activate when I'm at the interface of fat and water. Now, to work at the interface, you need mixed micelles of phosphatidylcholine and bioacids and another enzyme called collapsase. This is a 90 residue protein, and it forms a 1-to-1 complex with lipase. So these two will interact together and work together to digest lipids. Now, lipase, the active site, is in the N-terminal domain, and the catalytic triad of lipase actually looks a lot like the serine proteases we've talked about previously back in Biochemistry 1. Now, if there's no lipid for phospholipase to work with, the active site is covered by a lid, which is just a 26 residue cover. Once our lipase comes in contact with a micelle, what will happen is that the lid will reorganize, move out of the way, and expose the active site of our enzyme. And our enzyme will have a hydrophobic surface near the active site, which makes sense. This enzyme works on lipids, so the active site should be hydrophobic to help and get these lipids into the active site. Now, collapsase will bind to the C-terminal domain of lipase, and it has hydrophobic tips on 3 different loops that will extend from this complex. And this is kind of like a binding platform, a hydrophobic binding platform that will help to bind to lipids. And overall, it just stabilizes our lipase and lipid conformation. Now, if we want to look at a specific lipase, we can our model is going to be phospholipase A₂, which contains this hydrophobic channel that we said previously. And this allows phospholipids to be fed right into the active site. Now, once we have broken down the lipids in our small intestine, they're going to be absorbed by the cells of the small intestine, our intestinal mucosa. And for the absorption, bioacids are needed to absorb these into our cells. These bile acid micelles allow the non-lipid portions to be transported. You also need these if you want to absorb vitamins A, D, E, and K. Now, once absorbed in these intestinal cells, our fatty acids are going to form a complex with the fatty acid binding protein IFABP, intestinal fatty acid binding protein. Finally, an enzyme name that makes sense. And this protein is found in the cytoplasm. So what this IFABP does is it increases the solubility of the insoluble parts of our fatty acid, and we're also going to protect ourselves from the detergents of the bile acids, because remember, our cell membrane is polar and nonpolar, and a detergent would actually destroy our cell membrane, which would be bad because we'd leak out everywhere. So we're helping to protect against these bile acids and fatty acids. So here our IFABP has 10 anti-parallel beta strands, which is in 2 beta sheets as shown here. There's a gap in these strands where fatty acid will fit, and here are fatty acid in the strand. The head group of the fatty acid will interact with arginine, glutamine, and 2 different water molecules of our IFABP, and the tail is going to be carried around several hydrophobic residues. And you can see this picture is actually taken from Texas A&M University, so not too far away from us. Once we have our fatty acids digested in our cells, then we need to transport them around to the body, and we transport lipids in complex with proteins. They're called lipoproteins. They're globular micelle-like particles, where at the core of our lipoproteins are our triacylglycerols and cholesterol, and on the outside we have amphiphilic coating of proteins, phospholipids, and cholesterol, so this can be water soluble. We have different types of lipoproteins. So we have the chylomicrons. They're going to circulate around the blood to deliver our dietary triacylglycerols and cholesterols. Then we have very low density lipoprotein, intermediate density lipoprotein, and low density lipoprotein. These lipoproteins are synthesized in the liver, and they transport internally produced tags, so the tags we make from the liver to the tissues, while high-density lipoprotein is going to transport cholesterol and other lipids from our tissues back to the liver. And these different densities, basically, as our lipoproteins become smaller, the density increases. So HDL is the smallest because they have the highest density, and VLDL is the biggest because they have the lowest density. So let's look at LDL here. So the protein components of our lipoproteins, if we're just looking at the proteins, we call them apo or apolipoproteins and apoproteins. And you have about 9 different apolipoproteins, and they're just distributed in different amounts in our different lipoproteins. One type of lipoprotein is called ApoB100. That's going to be with LDL, and it's extremely hydrophobic. So every LDL molecule you have will have one ApoB100. That's what this yellow line is here. That covers roughly 50% of our lipoprotein. Other lipoproteins that we have in our body are water soluble, and they'll weakly associate with our lipoprotein here. And there are a lot of helixes, which makes sense. One part of the helix will be polar, one part will be nonpolar, so we can interact both with water and the lipid. Now, if we look at another apolipoprotein called A1, that's going to be in your chylomicrons and HDL. Here is just a 22 residue repeat of similar sequence. So we have a nonpolar sequence and a polar sequence, and every so often you're going to have a kink at proline. So every 22 residues you have proline, which allows this coil to curve. And you have 4 monomers associating together as seen here. So we have purple, blue, yellow, and green all coming together. And this will help wrap around HDL. So the nonpolar will interact with the lipid, while the polar will interact with water. And that's it for this video. I will see you in the next one.